We'll wait for a few minutes, 2 3 minutes, and then we'll start the session. Okay. Good afternoon, everybody. Welcome to the sixth live interaction session. So this week I'll be discussing MCQs related to week four's content, and similar to the previous live sessions. Uh, I'll first go through the MCQ. I'll give you guys around 30 seconds to type your response in the chat box, and then I'll go ahead and discuss the MCQ question in detail. So I'll get started now. I hope my screen is visible. Kindly type your answer whether it's visible or not in the chat box. Okay. Okay. So uh, in week four we were introduced to various concepts uh, such as peak EMG, muscle spindles, proprioceptors, spinal cord injuries, and various other things. So the first question is, which of the following sequence specifies the correct order in EMG signal processing? So there are four options. I'll give you 30 seconds, and you can give a response in the chat box. Okay, let's see. Any response? C. Okay, Monojit has responded C. Okay, let's discuss this now. So first, we need to get data, get the EMG data from the electrode. So we need to decide a sampling rate at which we are going to get data. So typically, an EMG signal, the information of an EMG signal is between 20 hertz to around 500 hertz. So we have to first sample the data at a frequency of about 1000 hertz typically, so that we don't lose out any information. So first is sampling. After we sample the data, we need to filter the data to remove any noise. So the next step is filtering. Then the third step is uh, we now need to process the data to get some kind of information out of it. So one of the steps to this is rectification. And finally, the last step is integration. So this is the answer is C. I think Monojit got it correct. Uh, so let's have a look at the slides as well. What is there in the slide? So we can see that there are various kinds of EMG. There's surface EMG, there's needle EMG. So we can see here that this is how the EMG signals are processed. First there is filtering, then there is rectification, and then there is integration. But even before filtering, we need to get the data. So we need to sample it. So these are the steps. Uh, so these are the various stages. First is sampled at a high frequency. The second is filtering using filters. Third is full wave rectification here. Fourth step is smoothing. So this is we can either integrate or we can after rectification there are various steps. It need not be only integration. We can calculate the root mean square value. We can do other processing techniques to get some information out of the data. So here it is written. Integrating a rectified EMG gives a value that reflects the total current as well as the total resistance between the electrodes. Okay, so now let's go on to the next question. Free nerve endings is a type of A. Photoreceptor, B. Mechanoreceptor, C. Nociceptor, or D. Chemoreceptor. So you can take 30 seconds and then we'll discuss this question. Okay, let's see any response. Okay, Monajit is saying D. Let's discuss this. Okay, chemoreceptor. Uh, okay, so a better way to do this is we can get, let us go and look at the slide. And so we have receptors part 2. Okay, so you can see here that so there are various receptors. There are articular receptors which are in the joints, and then we have cutaneous receptors which are on the skin, and there are various types of these cutaneous receptors. So we have nociceptors, which is for pain. Then we have mechanoreceptors, which is to sense haptic feedback. For example, we can sense pressure, vibrations, and all that using mechanoreceptors. Then we have temperature sensors, uh, thermoreceptors on in the skin. So here, what you are seeing, these are various receptors. These are mechanoreceptors. So there are various types of mechanoreceptors. So there is Meissner corpuscle, then we have Pacinian corpuscle, we have Ruffini endings, we have Merkel discs. Then we also have free nerve endings. Right. So we'll just look here. Uh, so here free nerve ending is associated with pain and itch. So this is a nociceptor, and so the answer is free nerve ending. So let's go back and see what the options are. So the correct option is C. These these are not the options. Correct option is C. Answer is C. Nociceptor. Uh, Going to the next question. Proprioceptors have antidromic conduction, orthodromic conduction, both antidromic or and orthodromic conduction. D. Neither antidromic nor orthodromic conduction. So I'll give you guys around 30 seconds again, and you can try and answer this question. Okay, let's see what the answers are. Monojit is responded with C. Okay, okay. Yeah, that is the correct answer. The answer is C, both antidromic and orthodromic conduction. So let us first have a look at the concept of proprioceptors. So we know that, let me draw a proprioceptor. So this is a cell body of a proprioceptor, and we know that it has two axon terminals, right? And this is the sensory ending. So basically, proprioceptors are used to take sensory information uh, uh, from various modalities, of different modalities, to the central nervous system. So such type of axons, which or nerve or or neurons, which take information, sensory information, uh, uh, which send sensory information and take that information to the central nervous system, are called afferent neurons. So proprioceptors are afferent. So this is an important point that you need to remember. Afferent. And those neurons or axons which uh, bring motor commands to the muscles are called efferent neurons. So motor neurons are E, EFF, ERENT, efferent neurons. So this is something that is important that you need to remember: efferent and afferent. So coming back to the proprioceptor, which is an efferent neuron. So we know that the cell body of a proprioceptor is actually located in a ganglion. So a ganglion is a collection of cell bodies. So the cell body of a proprioceptor is located in a ganglion which is close to the spinal cord. Okay, so this is a ganglion which has many other cell bodies, and we have a spinal cord here. So the cell body is located close to the spinal cord, and it has a characteristic T shape. So one end goes to the periphery of the body, and its ending the ending of this axon is has a sensory ending, so it can sense various type of modalities. It can be force, it can be length, it can be pain, it can be temperature, it can be it can be mechanically sensitive. This is detected by the sensory ending, and then we have these signals which travel to the cell body. So this is called antidromic conduction. And then some sort of processing happens here, and then we have another set of action potentials or signals that travel to the spinal cord. So this ends synapses with spinal with neurons in the spinal cord. Uh, so this is called orthodromic orthodromic conduction. So conduction from the sensory ending to the cell body of proprioceptor is called antidromic conduction, and conduction of signals or action potentials from the cell body of the proprioceptor to the spinal cord is called as orthodromic conduction. So you got it right. The answer is C. Uh, Moving on to the next question. Which of the following is true for a muscle spindle? One muscle spindle is made of. Okay, so I'll give you guys around 1 minute to answer this, uh, and then I'll discuss this in detail. Okay, let's look at the answer. Okay, Monojit is saying D. Okay, let's discuss this. So the Monojit's answer was D. Which of the following is true for a muscle spindle? So let us recall that muscle spindle is one of a very amazing uh, uh, system which is in our muscles, which senses uh, the velocity of stretching of the muscles as well as the length of the muscle. So from the length of the muscle, we can the central nervous system can get an idea of the first of all the joint angles as well as the relative orientation of the segments relative to each other. So this is a very important uh, system, muscle spindle, a very important sensor. So you need to know everything about a muscle spindle. You can expect questions from this particular uh, uh, related to this particular concept in your exam. So let us go through these options. One muscle spindle is made of 2 to 3 chain fibers and 5 bagged fibers. I think this is wrong because I think it's made up of 5 chain fibers and 2 to 3 bagged fibers typically. So this is not the correct answer. Muscle spindles are made of contractile central regions and non-contractile polar regions. This is again false. We know that a muscle spindle has an intrafusal fibers whose central region is non-contractile. Okay, and the polar region is contractile. So the central region is are innervated by proprioceptor sensory endings. So the primary sensory endings and the secondary sensory endings, and the polar regions are actually contractile and they are innervated by gamma motor neurons. Uh, so this is again the wrong answer. Nuclear chain fibers are divided into static nuclear chain fiber and dynamic nuclear chain fiber. No, this is wrong. So it is the nuclear back fibers that are divided into static nuclear back fibers and dynamic nuclear back fibers. So this is the wrong statement. D. The central portion of the back fibers are innervated by afferent neurons, whereas the contractile portions are innervated by efferent neurons. So yeah, this is correct. So we know that a muscle spindle has these intrafusal fibers which are parallel to and this is surrounded by extrafusal fibers. Okay, it is surrounded by extrafusal fibers and the central region is non-contractile and polar regions are contractile. So these are innervated by gamma motor neurons. So there are two types of gamma motor neurons: the static gamma motor neurons and dynamic gamma motor neurons. Similarly, the middle region, the central region, is innervated by uh, uh, sensory endings of proprioceptors. So there are primary sensory endings and then there are secondary sensory endings. So primary sensory endings have belong to 1A type of axons, whereas the secondary sensory endings belong to the category 2 axons. Uh, so there is a hierarchy. There are different kinds of intrafusal fibers as well. So we won't get into that now. Perhaps in a future question we'll encounter this. So the correct answer is D. So yes, the answer is D. Okay. Moving on to the next question. Question 5. Alpha gamma coactivation helps in. So again, this is an important question. This concept itself is important. I'll give you guys around 1 minute to answer this, and then we'll go through this together. Okay. Monojit is saying C. Okay, let's discuss this. Okay. Monojit's response is C. Okay, so we know that alpha gamma coactivation is a very important concept. So first, let us look at this concept. So there are two types of gamma motor neurons. One is the dynamic gamma motor neuron, which innervates the dynamic back fibers. So we know that the intrafusal fibers. Let us just go through that hierarchy. So we have muscle spindle has. Let me just create some space. Muscle spindle has intrafusal fibers, and there are many types of there are three types of intrafusal fibers actually. One is nuclear bag fiber, and then we have nuclear chain fiber. So the name why it is like this is because of the I think the way the nucleus is arranged. I think uh, so. In the case of the chain, it is arranged in chain fashion, whereas in bag it is clustered. In the nuclear bag fibers, it is clustered uh, like a bag. Hence the name nuclear bag and nuclear chain. Then we have two. There are two types of nuclear bag fibers. One is static bag fibers, and then we have dynamic bag fibers. Okay, so we have a single 1A large axon uh, proprioceptor. So we have, like we discussed before, we have proprioceptors which innervate these muscle spindles. So again, there are two types of proprioceptors. One is primary. One has primary spindle endings or primary sensory endings, and these innervate all three types: static, dynamic, as well as nuclear chain fibers. Uh, and the axons of these type of primary sensory endings are called as 1A. 1A. So these are the proprioceptors, 1A proprioceptor, and then we have another category. So the axons of these type of proprioceptors, the 1A proprioceptors, are larger and their conduction velocity is faster. Whereas in type 2 proprioceptors, the axons are smaller compared to 1A, and the conduction velocity is also smaller. However, these innervate the sensory endings of this innervate static bag fibers and your chain fibers. Okay, so this is one thing that you need to keep in mind. So this is how the structure is arranged. Apart from this, we have gamma motor neurons, which innervate the polar regions, like we discussed before. So there are two types of gamma motor neurons: dynamic gamma motor neurons, which innervate the polar regions of the dynamic nuclear bag fibers. Okay, and then we have the static gamma motor neurons, which innervate the static as well as the nuclear chain fibers. So this is the arrangement of a muscle spindle. So now, keeping all this in mind, let us discuss this. The alpha gamma coactivation helps in keeping the polar region of the extrafusal fiber stretched when the intrafusal muscle, when the intrafusal fibers contract. No, first of all, we know that. Let me just go to a new slide. So there's no space here. Let me just duplicate this. Control C. Let's erase this. So this is a tricky concept. Muscle spindles has many factors, many things associated with it. It's tricky to remember this, but once you hang off the hierarchy, the way what is innervating what, then it's a cakewalk. So what happens is we have the intrafusal fibers here, and we have the extrafusal fibers. So these are the extrafusal fibers, let's say. So when the extrafusal fibers contract, since the intrafusal fiber is also connected, the ends of the intrafusal fiber is connected to the extrafusal fiber or to the tendon. So when the extrafusal fiber contracts or lengthens, let's say this extrafusal fiber lengthens, then even the intrafusal fiber lengthens, and then the sensory endings here of the proprioceptors are sensitive to this stretch, and hence there is a signal that goes to the proprioceptor via the proprioceptor to the central nervous system. So this is the basic working. Now what happens is, let us say that this is at a resting length, right? This is at a resting length. Uh, since this is at a resting length, this particular intrafusal fiber, so this is the extrafusal fiber, and this is the intrafusal fiber. So at resting length, what happens is this is slack. This is not. This does not have any tension. So when this starts to lengthen, there'll be a time. There'll be a point where this does not immediately feel the tension. So there'll be from the resting length. There'll be a certain amount of stretching which will not be detected. So for that, what happens is we have a gamma motor neuron, uh, which is innervated at its extremes here, at the extremes of the extrafusal fiber, and when this contracts, so when the muscle is at a resting length, the gamma motor neuron contracts and keeps this particular the middle region where the sensory endings of the proprioceptors are. Uh, uh, it keeps it taut, or it keeps it stretched. So whenever any external stretching happens, immediately what happens is this is sensitive to that, uh, to the external stretches at all times. So that is the functioning of the gamma motor neurons. So the information from the firing information that is required to keep this particular intrafusal fiber sensitive or taut, uh, plus the information from the sensory endings. This is both used to get a sense of the length of the muscle fiber. So whenever the alpha motor neuron actually fires, even the gamma motor neuron fires to keep this particular the central region sensitive to stretches. So this is the alpha gamma coactivation. So if we go through this, keeping the polar region of the extra fusal fiber stretched when the intrafusal fiber contracts, we know that uh, this is not correct. We just discussed this. Keeping the contractile region of the intrafusal fiber stretched when the extrafusal fiber contracts. Keeping the central region of the intrafusal fiber stretched. This is correct. Keeping the central region of the intrafusal fibers stretched when the extrafusal fiber fibers contract, or keeping the polar region of the intrafusal fiber contracted when the extrafusal fibers stretch. This is correct, right? So when the polar region should be contracted when the extrafusal fibers stretch. So this is the correct answer. Answer D. Let us also discuss answer C. Why C is wrong. Uh, Keeping the central region of the extrafusal fiber stretched, of the intrafusal fiber stretched. This is correct. When the extrafusal fibers contract. Uh, so in this case, I think even this is correct because. Uh, so I think both these answers are correct. The answer should be C as well as D. Uh, Contraction uh, of the extrafusal fibers will also stretch the intrafusal fiber, right? Because we know that intrafusal fibers are connected to the tendons as well. So tension in the tendon will tend to stretch the intrafusal fibers. Uh, Information from the central region as well as the the contraction that happens of the extrafusal fibers, which causes the intrafusal fiber to stretch. So this both this uh, together will give the total length of the muscle. So I hope this explanation was clear. Uh, Monojit, I hope you understood this, what I explained just now. So the answer is C as well as D. Okay. Okay. So Golgi tendon organs are located dash and act as dash sensors. So again, I'll give you around 30 seconds to answer this, and then we'll have a discussion. Okay, let's see what the answer is. A. Okay, Monojit has responded with A. Okay, let's go through this. So let us recall that Golgi tendon organs are force sensors. So we have a muscle here. Let's say we have a muscle here, and we know that uh, the tendon. So this is the tendon. This is the muscle tendon. We know that the tendon connects the muscle to the bone. So this is the bone. Let's say. So what happens is uh, Golgi tendon organs are actually force sensors. They are very accurate force sensors. And if you zoom in here at the junction between the muscle and the tendon, that is where the Golgi tendon organs are connected in series. So we should recall that muscle spindles are connected in parallel to the muscle fiber, whereas Golgi tendon organs are connected in series to the muscle fiber and the tendon. Okay, so what happens is if you recall the structure, you have these collagen fibers. So this is your Golgi tendon organ. You have various collagen fibers here. Okay, and you have a proprioceptor. The sensory endings of proprioceptor come in here into the Golgi tendon capsule, and these are actually one B. This is a proprioceptor, 1B proprioceptor, and it comes and intertwines with these collagen fibers. Okay, so its sensory endings intertwine with these collagen fibers. When the collagen fibers, when uh, the the tendon and the muscles stretches, when the tendon stretches during contraction, what happens is this collagen fibers straighten out. Okay, once they straighten out, the sensory endings between the collagen fibers that are intertwined between the collagen fibers, they get pressed. Okay, and when they get pressed or they get squished in between, when the collagen fibers straighten out, then they start generating action potentials, which are then sent to the central nervous system. Let us have a look at how it looks. Where is this Golgi tendon organ? Yeah, so you can see here how the collagen fibers intertwine. Uh, these are the collagen fibers, and then the 1B afferent. Uh, the sensory endings of the 1B afferent come and intertwine with this. So you can see here, and once this stretches, it gets squished. The 1B sensory endings get squished in between, and then it generates action potential. So this is how it works. So let's go through the options. Are located between primary and secondary muscle spindle force, muscle spindles, between tendon and muscles. So the answer is actually not between extrafusal and intrafusal fibers. Like I just explained, the Golgi tendon is between the tendon and the muscle, and it measures force. So the answer is D. Between tendons and muscles, but this is also correct, but it does not measure length. So the answer should be D. Moving on to the next question. The sensory receptors in your skin that report changes in pain and itch are. So I think this is a repeated question, but we'll go through it anyways. So again, I'll give you 30 seconds to answer this. Okay, so let's see what the response is. Typo problem. C. Okay. Yeah, there is a typo problem here. Pain, pain and itch. It should be and. The sensory receptors in your skin that report changes in pain and itch are. Uh, so let's see. The response is C. Okay. So let us just go back to that slide. So we can see here that pain and itch are nociceptors, and they are associated with free nerve endings. Okay. Uh, so again, this you should remember all these this particular table. You might expect certain questions from this in your exam. Uh, so this is an important part. Yeah, pain and itch are nociceptors, and it's associated with free nerve endings. So let's see. The answer is B. Answer is B. Which of the following is not a part of the extrapyramidal tract? Okay, again I'll give you 30 seconds and then we'll discuss this. Okay, 30 seconds up. Let's see. B. Reticulospinal tract. Okay, this is the response. Let us see. Let us discuss this. So we know that in the spinal cord we have various neurons that are traveling through the spinal cord, and then they come out through uh, the vertebrae, through gaps in the vertebrae, and also we have a lot of nerves that go through the spinal cord into the brain cell, into the brain. Uh, so we have two types these the different tracts. Uh, so a tract is basically a nerve that is a bunch of nerves that are traveling, or a bunch of axons that are traveling through the spinal cord. Uh, so we can divide these nerves into various tracts. So there are two types of nerves that are going the through the spinal cord. One is the descending tracts, and the other one is ascending. So the ascending tracts basically get sensory information from various parts of the body, sensory info to the central nervous system. So these are basically afferents, and then we have the descending tracts, or the motor, which bring motor info to the muscles. So these are the descending tracts. They get motor information from the brain to the muscles. Uh, this descending tracts can be divided into two parts. One is pyramidal, and the second is extra pyramidal. Okay. So the tracts that the neurons that uh, belong to the pyramidal actually originate from the cortex. So we have the cortex, and then we have other parts of the brain. So whatever originates from the cortex uh, in the descending pathway are comes under the pyramidal tracts, and everything else comes under the extra pyramidal tracts. So the neurons that connect the cortex to the spinal cord are called as corticospinal. It's called the corticospinal tract. So this belongs to pyramidal, right? So the remaining things belong to extrapyramidal. So rubrospinal tract, reticulospinal tract, vestibulospinal tract belong to extrapyramidal, whereas the corticospinal tract belongs to the pyramidal. So this is not part of the extrapyramidal. So I'll just open uh, the corresponding slide. So you can see here motor and descending. So this is the efferent pathways, and the sensory endings are the sensory pathways are the efferent or the ascending pathways. So we have two types of motor tracts. One is the pyramidal and the extrapyramidal. So whatever is connected to the cortex. So here lateral corticospinal tract, anterior corticospinal tract comes under pyramidal, and everything else comes under uh, extrapyramidal. So the answer is C. Going on to the next question. GABA neurotransmitters causes. Again, I'll give you 30 seconds and then we'll discuss this. Okay, let's see what the answer is. What is the neurobiological implementation of motor primitives slash motor synergies? Neurobiological implementation of motor primitives slash motor synergies. Monojit, from which part of the course are you referring this question with? Is it related to this current weeks, or do we see this in the future? Or is it just a generic question that you're asking? Okay. Uh, so I am not sure whether I completely understood the question, but so we have various muscles in our body, and these span across various joints. For example, uh, we have let us consider the biceps, for example. So the biceps. Let me just open the camera. So these are my biceps. So when the biceps contract, the contraction pulls causes flexion, right? Now what happens is, uh, so we know that the biceps is involved in flexion, and the triceps, which is here, is involved in extension. Okay, but the bicep is also involved in this action. So this is pronation, this is supination. Okay, uh, so if you do this, you can see, if you put your hand here, you can see that this is contracting. So a bicep is involved in both uh, uh, flexion as well as supination. So when I'm doing a supination action, and when this particular bicep is contracting, uh, what happens is I want to supinate, but the biceps is involved in both supination as well as flexion. So I need to prevent the flexion component of the biceps from. I want to prevent the flexion. So what happens is the triceps also contract. So during supination, uh, not only is the biceps involved, but there are supinator muscles, and there is also activation of triceps to prevent the action of flexion. Okay, so there is coordination that is happening. There is, so this is just one example. So there is a lot of coordination between muscles that is happening. So and this coordination is can be referred to as a synergy. Uh, how exactly the synergy happens? How what are the mechanisms that the brain? What are the strategies that the brain employs to coordinate these activation patterns uh, is actually an area of research. Uh, so, I mean, this is what I understand of when I talk about motor synergies. Uh, the exact mechanism as to how the brain controls these activation patterns, or how the brain generates these activation patterns, is actually still an active area of research. There are various theories, and there's still active research going on in this field. Uh, so I hope that somewhat answers that question, Monojit. Hello. Okay. I hope you were able to hear me. Uh, I'll now finish discussing this particular question. Okay, clear. Okay, Manojit. Now I'll just discuss. Let's discuss this particular question. GABA neurotransmitters causes a postsynaptic excitation by attaching to nicotinic cholinergic receptors. We know that this is false because acetylcholine attaches to nicotinic cholinergic receptors. Then we have B, postsynaptic inhibition by attaching to ligand-gated or metabotropic receptors. Yes, this is correct. Uh, so we know that when it attaches, the GABA attaches to these ligand-gated receptors. Uh, what happens is there is either two things can happen. Either there can be an outflux of potassium, or there can be an influx of chlorine ions. Uh, so these two are the things that are involved, are the receptors that are involved. So then presynaptic, it does not. So GABA neurotransmitter does not have anything to do with presynaptic. So these two options are anyways eliminated. So the answer is B. Uh, it's about 6 o'clock right now. We'll stop the session here. And yes, yes, Manoj, your option was B. Correct, you got that right. So we'll continue the discussion from next week. And regarding to your question as well, there is a very good book called Synergy, which is written by Mark Latash. So that's a very good book, and it's an easy read. So you can refer. You can also read that book if you get the time. So our the instructor of this course is Vardhan SKM. His PhD advisor is Mark Latash, and he has written this book called Synergy. It's a very good book, and I think you can, if you get the time, you should go through it. Uh, okay, Monajit, thanks a lot for joining in to this session. Uh, Kindly join in next week as well. See you next week. Bye.